Thank you very much, Jane, for introducing me this afternoon. Good afternoon to all of you. Good morning, good evening, wherever you are, at what time you are. I'm very glad that you are here with us today. And today I'm going to present the highlights of our year 2021 at the Food Packaging Forum. So we have been working on human health and environmental impacts of food contact articles and their chemicals. In the beginning of my presentation, I would like to show you an overview of 3 ongoing research projects. The first one on this list is an EU-funded project named Aurora that focuses on early life human health impacts of micro and nanoplastics. You will hear more about this project in the presentation by Juliette Legler later on this afternoon, so I'm not going to give you any more details on this project. The second one we are currently working on and are involved in is the Understanding Packaging Scorecard. In this project, a tool is being developed that helps making informed decisions when choosing food packaging and footwear for a defined application. And also here you will have the chance to hear much more about it on Friday afternoon in the presentation by Florian Sutter, my colleague at FPF. The third project we are currently working on is the Food Contact Chemicals and Human Health Project, so FCCH project as we call it. And this one I'm going to present in more detail today. So it deals with food contact chemicals and how they could affect human health, and we really want to map all the food contact chemicals in a systematic way and address also these impacts on human health in a systematic way. So here you see, again, an overview and very importantly, some definitions. I know that many of you have heard these definitions quite some time already, but since I'm going to use the acronyms again in the following slides or over and over again in the following slides, I would like to quickly explain these abbreviations. So the first one we are usually talking about is the food contact chemicals, FCCs. And if we talk about FCCs, we mean all the chemicals that are on the one hand used to manufacture food contact materials and food contact articles. And at the same time, we also include all the chemicals that may be present in the final article. So FCC, the term covers both the intentionally added substances and the ones which are present in the final article. Then food contact materials, that's quite obvious. That is that are all the materials that can be used to manufacture items that come into contact with food, for example, plastics, paper, and board, typical for packaging, but also metal, glass, and also some smaller or not really, structural materials such as adhesives, printing inks, coatings, and so on. And using food contact materials, you can produce a lot of food contact articles. Either you use one material, for example, if you have a wine glass, you typically have just glass. Or if you produce a yogurt cup, you may have quite some different food contact materials involved to produce the final food contact article. So we are using these terms frequently, and that's why I listed them here. Okay, I mentioned it briefly before. In this FCCH project, we want to map the scientific evidence on FCCs and human health. And to have a foundation for this work, we first of all started to compile a database on intentionally added FCCs. And this database, we called it the FCCDB, that has been published already last winter. And it contains 12,000 chemicals that may be used during the manufacture of FCMs and FCAs. We don't know exactly which of these 12,000 chemicals are used, but we really compiled them from published and regulatory lists. So they appeared on this list because of their potential being a part of an FCM or FCA. And you can find much more information about this database either on our website or in the scientific publication. Now, in the next step, we would like to map the scientific evidence for the other three topics you see here in this illustration. And currently, we are working on the first scientific evidence map. This one focuses on migration and extraction of FCCs from FCAs and FCMs. And we want to systematically answer the question which FCCs have ever been measured in migrates and extracts of FCMs and FCAs. And the product will be, again, another database that we are planning to share with you in the near future. And the other two parts you see here will also be addressed in a systematic manner. And we want to answer the question, what of these FCCs that are either intentionally added or migrate or have been extracted from FCMs and FCAs have been found in the human body, in human biomonitoring studies, and how these chemicals could affect human health. So I already told you the aim of this second project part is a database, the FCC MIGEX database. And the database will focus on FCCs, and we really want to collect as many FCCs as possible from the scientific literature in a systematic way. And for each publication which we looked at, we asked ourselves the same question. So which FCCs were analyzed and are reported in this publication? And that is, of course, not really giving you a lot of information. So we asked some more questions related to this FCC. For each FCC, we wanted to know whether the experiment that has been performed or described in the publication was a migration or an extraction experiment, and whether migration into food or food simulant was measured. Then we wanted to know what kind of FCA was tested, what kind of FCM was tested, and whether the FCC that was measured was actually detected, yes or no. And that is our core information, which we compiled for many scientific studies and other reports. And here is one slide showing you how we really ran, or how our procedure was. So we started with a systematic literature search. So we defined exactly how we want to search the scientific literature and the gray literature, which terms we are going to use, and we came up with a list of 16,000 publications. And then we systematically screened all these publications in a two-step process, first based on title and abstract, and then on full texts. And afterwards, we had 1,200 records left that we really included for data extraction. So all the questions you have seen on the slide before were asked for each of the publication. And we compiled a lot of data with the help of some software tools. So first of all, we used Kadima for the screening, TechTalk for reading the paper that gave us support. Then we developed a tool, SciExtract, to really handle the data and to compile them in a systematic way. And after we have finished reading and extracting data from 1,200 records, we also needed to do some post-processing steps of the data to finally have a database or generate a database with more than 20,000 result entries, including more than 2,800 food contact chemicals that were detected in extracts or migrates of FCMs and FCAs. So here you see already the key data or the key information about our database. One of the first questions we asked ourselves was, now we know which chemicals are intentionally added to produce food contact materials because we have the FCCDB with 12,000 something chemicals. And now we also know which chemicals migrate or can be extracted from FCMs and FCAs. So we know that there are more than 2,800 FCCs. And something important I forgot to say when I talk about numbers, these numbers are always related to unique CAS IDs. We cannot easily count all the other chemicals because we cannot see, at the moment at least, if they belong to this, or if different names belong to the same group and so on. If you have been working with chemicals, you may know this problem. So these are only chemicals that have a unique identifier, the CAS ID, when I talk about numbers. So, but most of them have. So you see here, we have these two circles, and when we compared these two, we saw that the overlap is, I found it amazingly small. So there are 1,000 chemicals which are intentionally added and which have been measured in migrates and extracts, but there are 1,800 chemicals, which obviously are not on the list of intentionally added substances, but nevertheless, they have been found in migrates and extracts of food contact materials and articles. So we call this the universe of FCCs, both circles together, and there are more than 14,000 chemicals on these two lists now. When we sort these chemicals per type of FCM, the FCC MIGEX chemicals, we see that most of them have been measured in plastics. A few of them, or still almost 1,000, have been measured in paper and board. Multimaterials is also a type or a group of FCMs that included many different chemicals, followed by metals. And here I have to say, there many of the chemicals seem to originate from the coating. And then glass and ceramics is having the lowest diversity of chemicals. And this group of others includes, for example, FCMs such as wood, silicones, rubber, textiles, and so on. So this is quite a diverse group, maybe explaining that the number of chemicals is also pretty high here. We also, among the many questions we could ask, we also generated a list of top 5 FCCs per type of FCM. And you don't need to see or look at the details here because, yeah, we wouldn't have enough time to explain all of them. But if you look at the colors, you see something interesting. So the red color indicates phthalates. So you see that phthalates have been found in plastics on place 1 and 2, also frequently found in paper and board, frequently found in the multimaterials, and also in the group of other FCMs. So they seem to be in many FCAs. In contrast, metals have been found in metal packaging, quite not very surprisingly, and also in glass and ceramics, which is also not very surprising. But the list of the top 5 in metals is led by bisphenol A. So you find bisphenol A very frequently in metal packaging because of the coating. And you find bisphenol A on place 3 of the plastic charts. So these are the, maybe not big surprises, but showing the most investigated chemicals. And as I said, we have a lot of data now, and we could ask so many more questions, and I could explain you so much more about what we already found and see in this database. But since I don't have a lot of time, I just continue now with the further FPF activities. So here on the slide, you see the links. You don't need to read them now, but if you want to come back, you can always do so. So in the last year, we did not only work on scientific projects, but we also published a fact sheet or 5 fact sheets on food packaging materials and recycling. And in these 5 food packaging material fact sheets, we present information on application, material properties, chemical safety, and end-of-life options in a rather easy to understandable way. And we also focus on recyclability and how recyclability could impact the safety of food packaging materials. Then we also published a brand and retailer initiatives database that lists over 400 voluntary initiatives and commitments by food brands and retailers. And you're welcome to check the details here. I see I'm running out of time, so I will be really quick, but you have the link, and it's a great and searchable free tool available on our website. We published a video blog, Unwrapping Food Packaging is the title of this video blog, where Jane Munke presents fundamental topics and new scientific developments in food packaging and health. 4 episodes are already online, also here. Just come and have a look. And last but not least, the news. So we have published almost 1 news article per day since last October. And in total, we have more than 4,400 news articles that are still all freely available. So if you have a question, just come and have a look, or register for the newsletter, then you get it into your mailbox. And that was my last slide. I thank you very much for your attention. And I also have to thank our team members, all project partners, collaborators, our foundation board and scientific advisory boards, and all the donors. Thank you very much for your support. We are very grateful that you're with us and help us with our daily work. Thank you.